So good afternoon. We have already mentioned these technical standards as one of the first CGC joint projects with another organization, ACMG in this case. And these technical standards have already been published in the main issue of Genetics in Medicine. So hopefully many of you have already have a chance to read this manuscript, maybe even to start implementing some of the recommendations from these new technical standards. However, we still thought that it would be very useful to discuss the content of this manuscript, and especially to show some examples how some of the recommendations can be implemented on real cases from our laboratories. And we really hope that that will help with further implementation of these recommendations. And these recommendations are only valuable if they're being implemented in our laboratories and if they're helping us to improve interpretation and reporting of our copy number findings. So I'm presenting this on behalf of the working group that consisted of individuals who are listed here. This was a wonderful group of colleagues who worked very hard to put this together. So I would like to take the opportunity to thank everybody who participated in these guidelines. Especially want to thank our chair, Dr. Fadi Mikhail, who did a fantastic job keeping us on the task and making sure that we bring this project to successful completion. So when we started, what was available at the time were 2013 standards for chromosomal microarray testing in neoplasia. And those standards did address interpretation and reporting. However, only some broad principles were described. And those were more based on what we were doing at the time for constitutional copy number abnormalities. So they were not ideally tailored for somatic copy number aberrations. And in practice, interpretation remained very subjective and lacked standardization, and there were inconsistent practices between different laboratories. There was also a lack of coordination with sequence variant interpretation guidelines, which were developed between AMPASCO and CAP and were published in 2017. And as we know, interpretation of somatic copy number variants is more similar to interpretation of somatic sequence variant than maybe constitutional copy number abnormalities. It should use some principles that are more related to what we do for somatic sequence variants. So having this in mind, we realized that there was a need for standardized, objective, and evidence-based interpretation and reporting of chromosomal microarray results in neoplasia. And the work group was formed between ACMG and CGC with the following goals. We wanted to develop recommendations for classifying acquired copy number abnormalities and copy-neutral LOH using evidence-based system. We wanted the system to use objective criteria, to be user-friendly and easy to implement, to address variant complexity, be flexible, and also to complement sequence variant interpretation system. And we also decided that within the scope of this project, we will include reporting recommendations. So we will try to define what are the components of the report and what is the best way to structure the report for clear communication of the findings. In terms of how we approach this task, this is a quick review of the methods we used. We did comprehensive literature review, especially focusing on some existing practice guidelines at the time, and we used experience and opinions from group members. We came up with the first draft, and then we asked for community input. So you might remember that at one of the previous CGC meetings, we had presentation of our initial drafts, and we asked for input from our community. Then we did testing of the draft recommendations using cases from our own laboratories, revised our draft, and then we posted the manuscript and the recommendations for comments for ACMG members. This was also approved by ACMG Lab QA Committee and ACMG and CGC boards. We implemented all the input that we got from the ACMG, CGC boards, QA committee, and the ACMG members, and that was finally incorporated in the final recommendations. So these are the core components, core elements within this manuscript and these recommendations. The most important component of these standards is a 4-tier categorization system that describes categories into which we recommend that copy number abnormalities and copy neutral LOH regions get classified. We also provide definitions of common terms that are used to describe copy number findings in tumors. And so this was something that was brought up yesterday during panel discussion about chromosomal microarray testing in neoplasia. So I want to point out that some definitions of commonly used terms are present in this manuscript. We also describe considerations regarding interpretation and reporting of both disease-associated as well as unanticipated clinically significant germline variants. So there were also some questions regarding how do we deal with germline findings when we do copy number analysis in tumor samples, and I want to point out that some of that has been addressed in these guidelines. And finally, as I mentioned earlier, we wanted to address reporting, so we provide some recommendations how to standardize reporting, which components should be included, and how the reports could be structured. What is also important to stress, and that these recommendations are not limited just for copy number abnormalities that we are detecting using chromosomal microarrays, but this can be used for detection of copy number abnormalities detecting by other methodologies in tumor samples. So if you are doing copy number detection through NGS, you can still apply the same standards and same classification categories as you would if you detecting these abnormalities by array. So moving on to describing the most important component of these recommendations, which is the 4-tier system to classify acquired copy number abnormalities and copy neutral LOH regions. So variants are classified into 4 tiers based on the level of evidence regarding their clinical significance, which can be diagnostic significance, prognostic, or therapeutic. And this was aligned with standards in guidelines for interpretation and reporting of sequence variants in cancer. So we are aware that more and more laboratories are simultaneously looking at copy number abnormalities as well as sequence variants in cancer using an integrated approach. So we wanted to align these two recommendations, two sets of standards as much as possible in our recommendations. So here are definitions of different tiers, which we recommend for classification of copy number abnormalities. Tier 1 would be variants with strong clinical significance, and we subdivided this tier into 1A and 1B. 1A would be acquired variants of the most significance. Those are the ones that either specify entities in LOH classification or are included into professional practice guidelines like NCCN and guidelines COG and others, or they predict sensitivity to FDA-approved drugs. We also recommend to classify germline pathogenic variants associated with predisposition to the cancer that's being tested into this highest tier of clinical significance. Tier 1B would be variants that are associated with that specific neoplasm and have prognostic or treatment significance as shown by a high or good quality evidence. There also should be expert consensus regarding their clinical associations, and this should be confirmed and reproduced by independent groups. The next tier of significance would be tier 2. For the lack of a better term, we just call this variants with some clinical significance. And these would be recurrent acquired variants that are associated with that particular tumor type that you are testing, but they're not highly specific, which means you may see the same copy number variant in some other tumor type. In tier 2, you would also classify those variants that have been shown to have some association with prognosis or treatment response, but there is only average quality evidence regarding that association. The third tier would be clonal variants with no documented neoplastic disorder associations. So those would be all acquired variants for which you know that they're clonal in the tumor that you are analyzing, but you otherwise don't have any other information regarding their contribution to that tumor. And often you would classify variants into this category based on exclusion. So if the variant does not meet criteria to be classified into higher tier 2 or 1, and then those clonal variants would end up in tier 3. And finally, tier 4 would be all the benign or the likely benign variants. Those are the ones that are common in the general population, and those you most likely would not be even reporting. So as I mentioned, we also defined level of evidence that you have to classify variants into each of these tiers. So for tier 1A, it's the simplest definition of the level of evidence. Tier 1A would be only variants that are either in WHO classification or existing practice guidelines, or there is FDA-approved drug based on association of that copy number variant with sensitivity of a tumor for that FDA-approved drug. So for tier 1B, we require high-quality evidence or good quality evidence, and then high-quality evidence would be defined as well-powered studies in the form of randomized controlled clinical trials, systemic reviews, meta-analyses, or large cohort studies with consensus between experts. And then good quality evidence would be defined as multiple, at least 2 smaller clinical studies in form of cohort and case control studies. And there should be reproducibility and confirmation by at least 2 independent groups. And then finally, for tier 2, we require that you have average quality evidence, which is defined as small case series or multiple case reports. And these definitions of levels of evidence were adapted from the Oxford Center for Evidence-Based Medicine definitions. To make this easier to understand, we also provide multiple examples of variants in different tiers, both for hematologic malignancies and for solid tumors. So this is just a small example from a much larger set of examples that we provide in the manuscript. So these are variants for B-lymphoblastic leukemia, and here for tier 1A, we'll list multiple copy number losses, so multiple deletions that result in abnormal gene fusions that define one specific molecular subtype in WHO classification, which would be PH-like ALL. Or towards the bottom of this table, you see things like hyperdiploid and hypodiploid BLL. Again, these are combination of copy number variants that define HW subcategories. What I want to stress at this point is that you will sometimes not classify copy number abnormalities in isolation, but you will classify patterns of copy number variants. And this is where interpretation of copy number variants in tumors differs from sequence variants. Sequence variants are always classified individually, but for copy number abnormalities, they sometimes individually do not have a meaning, and only characteristic patterns of copy number abnormalities have some importance. So we address this in this guidelines, and we recommend that sometimes patterns of copy number abnormalities be classified as a whole. Then in tier 1B examples, we'll list, for example, Icarus deletion, because there is a lot of good quality evidence that Icarus deletions in BLL have prognostic significance. However, this has not reached yet any professional guidelines, and there are no changes in clinical management for patients based on this finding. And so that's why it doesn't reach the highest tier of significance, and it's classified as tier 1B. And then in tier 2, we list examples of CDKN2A and B deletions, CTV6 deletions, SPEX5 deletions. So these are all copy number abnormalities that are recurrent in BLL. So there is some association with the disease that's being tested. However, there is no either prognostic or therapeutic significance at this time for variants in this category. So this is an example how we sometimes need to classify patterns of copy number abnormalities rather than copy number abnormalities individually. So this is a pattern that corresponds to IM21. This is very characteristic combination of copy number changes that you see on chromosome 21 in this molecular subtype of BLL. So in this case, you would classify all these copy number changes as one characteristic pattern rather than classifying each copy number change individually. And this is another example of the same concept. So this is hypodiploidy, actually doubled hypodiploid case. So again, you would not classify each copy-neutral loss of heterozygosity for individual chromosomes individually, but you would classify this as a pattern. This would be double hypodiploidy or hypodiploidy, which is associated with one specific molecular subtype in WHO classification. So moving on to the next component of these standards, I mentioned that we provide definitions of commonly used specific terms. So here is a list of some of the terms that we define in this manuscript in order to improve consistency between laboratories in how we use all these different definitions. So detailed definitions will be provided in our case examples that follow this presentation. But I want to stress that some of the terms that we had discussion about yesterday and there were questions about them, for example, amplification, those have been defined in this manuscript. We also included reporting recommendations to specify components and structure of the report in order to improve clear communication of the findings. In addition to defining components and structure of the reports, with of course leaving a lot of flexibility to laboratories to adapt this to their situation. We also have some recommendations how to interpret and report disease-associated germline findings, as well as unanticipated clinically significant germline variants. So again, there were questions about this during panel discussion yesterday, and some of the answers are listed. You will be able to find in the manuscript, and we will also show some examples after this talk. In terms of reporting recommendations, I mentioned that we describe different components that you may want to include in your report, as well as we suggest some structure that your report may have. So results is a very typical component of a report that we will all have. And we defined what should be maybe required components of the report, some that are recommended and some that are optional. The results can be reported either as a table or nomenclature string, and this is something that's also given as an option in the current ISCN. The second component that you will typically have in your report is interpretation. So we recommend including interpretation for all tier 1 and tier 2 variants, so the ones that have clinical significance. And then for clonal variants, tier 3 variants, you may or may not want to include some interpretation. You will include recommendations for follow-up testing as needed. For example, if you have germline variants that are associated in a tumor being tested, or if you have incidental findings, so germline variants that are not associated with a tumor that's being analyzed. It's very important to include references because we are doing now classification based on objective evidence, and those references will be objective evidence that you used for putting variants into a certain category. In addition to these key components of these technical standards that I reviewed, there are many other components of this manuscript that we will not have time to get into, but there are a lot of general and special considerations discussing some scenarios that you may run into as you are doing review and reporting of copy number abnormalities in tumors, databases and resources that you can use for interpretation, tables that include many more examples than the ones that I was able to show today. We also have some report examples to give you some ideas how you may want to structure your reports. And then there is there are other resources in supplementary material, including supplementary figures that will illustrate some of the definitions for terms like amplification, chromotripsis, interchromosomal complexity, and others. So in conclusion, we hope that these standards will lead to more standardized interpretation and reporting of CMA results in neoplastic disorders. But for that to happen, these standards really have to be applied and used in our laboratories. So we would really like to facilitate implementation as much as we can. So we plan to follow up the publication of these standards in multiple steps. We'll try to facilitate implementation through showing multiple examples how these standards can be used. We will be soliciting your feedback, so we want to hear from you what is working and what does not work in these guidelines, how easy and practical they are to use. And then based on your feedback, we are planning to reconvene the work group and do some updates and improvements in future iterations of these standards. We understand that this is the first version of these standards, and that there might be some room of improvement, so we want to hear back from you as you start using this in your laboratories. So with that, we will move on to showing some concrete examples how these could be used for real cases from our labs.